Okay, so since nobody freaking fruit flies. Since nobody responded to those announcements that I made with specific topics they wanted me to cover, um, I'm basically just going to go through the same topic review that I did last semester. And if you guys want me to elaborate on something more, just let me know. So this is a kind of general overview of a lot of the major topics. Again, we're going to follow this format here. We're going to start off with reviewing our eukaryotes, our eukaryotes, and our viruses. And then we will jump into the genetics stuff and then get into the metabolism stuff, which is our enzymes, redox reactions, and aerobic respiration. And then we'll give an overview of the immunology stuff. And I say viral, bacterial, fungal, protozoal, helminth, infection, and disease progression. I'm kind of going to be covering that more with the meta review that I'm going to do after we do the basic topic review. So I'm going to record it all as one video, but if you have to go, then go. Don't hang around if you don't have to. Um, because a lot of this stuff I've covered in the recordings that I posted in like the announcement that I had that said, um, more like final review stuff or whatever. And those links that I posted from the topic reviews and the meta discussion, it's pretty much the same stuff I'm going to cover here today. But if you'd rather hear me talk about it today than watch an old video, then that's what we're here for. Because otherwise, I'm pretty much going to cover the same stuff. But so yeah. If you have any other thing you want me to cover, then let me know. All right, so we're going to start off with, this isn't what I meant to click on. We're going to start off with our microbes, our pathogens, and basically for all of our microbes and pathogens. Oh, that was dotted line and I didn't expect that. All microbes and pathogens will be split into two categories. You are either cellular, you're made of cells, or you are acellular. You don't have cells as part of you. A cell is a membrane-bound unit. That can self-replicate. Whereas acellular microbes will be categorized as either viruses or prions. We'll get into the differences between those two here in a little bit. I'll remind you guys about that in a moment. But our cellular pathogens can be categorized into two categories. These will be the eukaryotes or the eukaryotes. Get this shorter line. Eukaryotes, another word for them, and you might have seen it in some of the older videos. They used to be called prokaryotes. It's the same exact thing. I was born and raised calling them prokaryotes. And so it took me a long time to switch to the terminology eukaryotes. And so you'll hear me call them either one. It means the same thing. The characteristics of the eukaryotes is that they have no nucleus. They have no true organelles. This will include our bacteria and our archaea. For the eukaryotes, these guys always have a true nucleus. It's membrane bound. And they have organelles. What are organelles? Organelles are membrane bound also. So a nucleus is an organelle. Structures in the cells that have a specific function. So some of them that we reviewed previously, we have the nucleus, which is there to house our genetic information. It is like the brain of the cell. It is, has all the information that is needed to make a new cell in it. So this is the nucleus where we house genetic info. You also call it the control center of the cell, right? It governs pretty much everything that's going to be happening in that cell. Another organelle would be the endoplasmic reticulum. Also known as the ER. And what this does, there's two different versions of the endoplasmic reticulum, the rough and the smooth. The rough ER has ribosomes for protein production. And the smooth ER is where we will make carbohydrates and lipids. All right. And then another one would be the Golgi apparatus. The Golgi is for packing and shipping things that we made from the ER. It's the warehouse. It's going to be distribution, basically. And then another one, which we all are pretty aware of, we seem to be able to recall that the mitochondria is the powerhouse of the cell. But don't forget what that means. All right, the mitochondria being the powerhouse of the cell means that this is where ATP production is done. Primarily, not just saying ATP production, but really this is the place where aerobic respiration occurs. Right, so those are the organelles that belong to our eukaryotes. And we could say another one might be the chloroplast, which is where photosynthesis happens, but there's other ones too, like the lysosomes and the vacuoles and other ones, but these are big picture ideas we're covering today. So coming back over to our eukaryotes, we want to cover a little bit of information about our bacteria specifically. All bacteria have an inner membrane that we also call the cytoplasmic or plasma membrane. Inner or cytoplasmic. This just contains everything in the cell, okay? They also may have a cell wall. Most of them have a cell wall. In bacteria, that cell wall is made of peptidoglycan. And some of them have an outer membrane, which those ones that have the outer membrane, we call them gram negatives, right? This is outside of that wall. I'm going to draw you little pictures. This whole unit, whatever you have, if you have an inner membrane and a cell wall and that's it, or if you have an inner membrane, a thin wall, and an outer membrane, or maybe you're a mycoplasma and all you have is an inner membrane and an outer membrane and no cell wall. Whatever your outer unit is, all of that together is called the cellular envelope. An envelope is always whatever your outer coating is, whatever your outer covering is. And so we can basically look at this at gram positive or gram negative. I'm going to zoom in a little bit so I can draw these in a little more detail. A little disproportionate, but that's okay. So they will have a genetic information in a circle. Always their chromosome is in circles. It's in one big circle. So that will be their genetic information. And then all of their ribosomes and whatever else the cell does is just floating around inside. All right, so this is our cytoplasmic membrane here in the black here. Inner membrane or cytoplasmic. That's what the black will be. All right. If you are gram positive, you got a thick wall. That was horrible. This will be the wall for the gram positive. If you are gram negative, you have a thin one. And if you're gram negative, you have an outer membrane. Make it a little thicker than that. All right. So this one is gram positive, and the one on the right is the gram negative. For our gram negatives, on that outer membrane, they will have little molecules on the outside. These are lipopolysaccharide, LPS, also known as endotoxin. All right. Come on, thing. Keep up with me. And some of our gram positives might have little waxes on their outer wall. And this would be, some of them have this, right? Wax. And that is called mycolic acid. So only some of them have that mycolic acid wax. Whereas in the gram negatives, all of them have LPS. If you are a gram positive that has the mycolic acid wax, then you are a Mycobacterium species. If you are a gram negative that doesn't have a wall here, then you are a Mycoplasma species. So they're pleomorphic. They don't have a set shape. What is making that noise? I'm in here all by myself and there's some sort of noise across the room. Okay. Anyways, so this is the gist of our eukaryotes and our eukaryotes. So next I want to get into more detail on our eukaryote groups because we had to know about the fungi and the protista and all that other stuff. All right, we'll start off with the fungi. These include our yeasts and molds. And they have ability to reproduce either by budding or by these asexual structures. They also can go through sexual reproduction, which we didn't cover much and which I'm not going to test you on, but I am going to potentially test you on our asexual structures, which was in our learning objectives, right? So our asexual structures, you are either a conidiophore, conidiophores, I want to be sure that I'm spelling that, make sure this is clear. Conidiophores are open structures. They look like we think of our penicillium or even our aspergillus. So this might be penicillium, aspergillus. It looked like a dandelion, right? Whereas sporangiospores, these ones are covered. And so they would be literally like if you had that dandelion structure, but it will have a covering around it, a membrane layer around it, and then those will burst open. Whereas these guys just sort of like gradually just leak off or always are kind of dusting spores. So that's how they will asexually reproduce. So that's a gist of some stuff we want to know about the fungi. For our protista, there were two different kinds of protista, the algae. These guys are our photosynthetic ones. And the protozoa. And these were the ones that we focused on with our studies in microbiology because these are the ones that can cause disease for the protozoa. Protozoa are always unicellular, whereas the algae can be unicellular or multicellular. For example, kelp that's found in the sea is multicellular algae. It's not a plant. It's actually an algae. It's multicellular. But protozoa are always unicellular, always single cell. And we usually categorize them based on their motility types. There's 4 different motility types for our protista. The first one will be the non-motile. We'll just start there. These are known as apicomplexans. An example of this that causes disease would be malaria. Sorry, give me just a second. All right. Sorry. Someone messaged me and I needed to answer it right away. Then we have the ones that have flagella. Now remember in bacteria can have flagella if they're motile, but protista, protozoa specifically here, the protozoas can have multiple ways of being motile, and these are going to be 3 of them. The flagella, the cilia, or the pseudopods that they can be motile with, but bacteria can only have flagella. Bacteria flagella work by rotating, and then the bacteria move by runs and tumbles. Basically, the thing is rotating one direction continuously, it'll run, and then when it switches directions or stops rotating, it'll tumble, and then that lets them change direction. Whereas with our protozoa, these flagella work in a whip-like motion. They're also covered by the membrane of the cell. And so there's much more control in their movements with the flagella here. All right, so examples that cause disease with the flagella will be Giardia and Trichomonas. Which we learned about Giardia in chapter 23, and Trichomonas we learned about in chapter 24 if you've gotten through that. Then we have protozoa that move by their cilia, which are tiny little hair-like projections on the surface of the cell. And so if your cell was shaped, I don't know, like this, who knows? And they have tiny little hairs. They don't have one big long flagella. They have tiny little hairs all over them. Okay. And the tiny hairs beat the surrounding liquid, basically moving it like in an oar-like fashion, constantly beating in their environment. And some example of, well, there's one example I can think of that has cilia, and it's not that one that you need to know, but it can cause disease, which is Balantidium coli. You won't get tested on it, but I'm just listing it because it can cause disease. It's not in our learning objectives, but it can cause diarrhea. There's so many things that can cause diarrhea. One of the biggest mistakes that people used to make in the past when they started taking this course is like, I would say, oh, patient has diarrhea. What do they have? And list a whole bunch of things. Any of them could cause diarrhea, but everybody always wants to just say salmonella. Don't fall for that, right? So we've learned in chapter 23 is basically all diarrhea. So be aware, there's a lot of things out there that can cause diarrhea. So Balantidium coli is one that has cilia that can cause diarrhea. I'm not going to test you on it, but hey, you might see patients one day that have that. Then we have our guys that have the pseudopods. Pseudopodia is the multiple of pseudopod. It's proper. And these are known as amoebas. And so these have pseudo being fake, podia being feet. These are the guys that move out like a portion of their cell. So they'll look like this, and they'll scooch out a portion of their cell to basically grab on to wherever the surface is that it's reaching for, and then pull the whole cell along like that. So these are the fake feet guys. And so some examples of guys that cause disease that are amoebas will be Naegleria fowleri, and acanthamoeba. These guys are associated with amoebic meningoencephalitis. Remember, Naegleria fowleri is associated with primary amoebic meningoencephalitis, and acanthamoeba is associated with granulomatous amoebic meningoencephalitis. Acanthamoeba is also associated with keratitis, which is inflammation of the cornea due to infection with this amoeba. All right, and so then finally, our last kingdom, fungi is a kingdom, protista is a kingdom, and then we have our kingdom Animalia. And in kingdom Animalia, the microbes that we were studying were called the helminths. And these are parasitic worms. To go over these guys, go away. These are all multicellular. They have organ structures in them, just like we do, organs and tissues. And they have complex life cycles. So our parasitic worms, also known as the helminths. They fall into the kingdom Animalia. They are animals like we are. So, all right. We can categorize our helminths into two categories: the roundworms and the flatworms. The roundworms are also known as, scientifically, nematoda. And our flatworms are known scientifically as Platyhelminthes. All right, so our roundworms, some examples of these guys. Pinworms. If you guys can't recall the scientific name for that from chapter 23, this was Enterobius vermicularis. Another example might be Ascaris lumbricoides, the giant intestinal roundworm. Obviously not an exhaustive list, but to start with, because these are the ones I bring up because pinworms are probably the most common human helminth infection here in the US, whereas Ascaris lumbricoides, the giant intestinal roundworm, is the most common roundworm infection in the whole world. So very important ones to be aware of. All right, so our flatworms can be categorized into two different categories. The tapeworms and the flukes. The tapeworms are known as cestota. An example of this one would be the typical tapeworm, one we always think of that has the segments in it, known as Taenia solium. All right, and then the flukes are also known as trematoda. Some examples of these would be schistosomes, like schistosoma mansoni. These schistosomes are the ones that are known as blood flukes. The blood flukes are the ones that get into intact skin in fresh water, and they have a life cycle in snails. So they're also known as it's like the snail disease or something like that. But basically, they have some sort of life step in snails, and it's due to contaminated water. So sewage and urine that is untreated into fresh water gets into the snails, and then the snails release a version of the schistosomes that can then get in through intact skin, causing swimmer's itch in people who go into the water. And then they are able to multiply in the bloodstream, causing a wide variety of issues in the spleen and the liver and the bladder. So another example would be our Chinese liver flukes. Those would be Clonorchis sinensis, for example. Yeah, so there's our helminths, all those covered in chapter 23. Okay, moving on. All right, now we'll come back around to our acellular pathogens. We'll start off with our viruses. Viruses are acellular. There's no cellular component to the virus itself. They rely on a host cell, and they will use the host cell's machinery to replicate themselves. And all they are really is some genetic information encased in some protein and maybe some membrane, right? And that's all they are. And then they just come into the cell, and the cell does all the work for them. So they themselves aren't cellular. And so because they are using all the resources of the cell against the cell's will, really, these guys are obligate. They have to. That's what obligate means. Obligate, intracellular, have to, inside of cells, okay? Parasites. Remember, parasite comes from our understanding of our symbiosis levels. So remember symbiosis, we had commensal, we had mutual, and we had parasite, right? And so commensal is where one benefits, the other one is neither harmed nor benefited. It just is, I don't know, being used by the other guy, but not at any harm. They might provide shelter for the guy or provide nutrients to the guy, but the guy that's housing him isn't being harmed in any way. Then we have the mutualistic symbiosis where both parties are benefiting, kind of like our gut bacteria. They're getting housed, they're getting nutrients, they're getting shelter, and we in turn are having our food broken down for us, and our immune system is being trained, and they're sending out signals to our body to help maintain our body and things like that, as well as providing microbial antagonisms, taking up the stadium seats and making antimicrobial peptides and stuff. So that's a mutualistic. We're both benefiting somehow. Whereas parasitism is where we have one organism being harmed and the other one is using that organism, causing the harm. And so viruses are doing that. So all viruses have a genome. They have two things for sure. The genome, which can be DNA or RNA. That DNA can be single-stranded or double-stranded. So can the RNA, which is unique because our cells can't have double-stranded RNA. In fact, this double-stranded RNA can serve as a PAMP in our innate immune system. That's so weird to us that we can immediately see it as foreign if it's there. All right, so if you're single-stranded, you could be positive sense, meaning that it could be read and translated straight as it is, or it's negative sense, meaning we have to make a copy of it in order to read or translate it. Same thing with the RNA. It can be positive sense or negative sense. So this is 6 different kinds of genome. Positive single-strand DNA, negative single-strand DNA, or double-stranded DNA. Positive single-strand RNA, negative single-strand RNA, or double-stranded RNA. 6 different types of genome, whereas cells always have DNA double-stranded. And I'll remind you guys, if you are a cell, all cells, we come back up here, all cells, that includes your eukaryotes, like your bacteria, and your eukaryotes, like your fungi, your protista, and your animalia. Bacteria, archaea, fungi, protista, and animalia are all cellular organisms, and they all have double-stranded DNA genomes, period. Their genomes are double-stranded DNA. So I just want to clarify that. All right. So you always have a genome. The other thing that all viruses have is a capsid. This is a protein coat surrounding the genome. And it's made of parts called capsomeres. All right. Some viruses have an envelope. This is a membrane that's stolen from the host cell, always. If they have an envelope, that is a membrane that they stole from the host cell. During the budding process, when they're pushing themselves out, they cover themselves with the membrane like a little blankie, right? If you are a non-enveloped, if you don't have an envelope, you're called naked. So that would include things like poliovirus. Poliovirus is a naked virus. There's a lot of, naked viruses out there but we think more commonly of the enveloped viruses like influenza, like COVID, like HIV, because of the disease that they're associated with. But there are plenty of naked viruses out there that can cause serious disease. So having an envelope or not having an envelope doesn't equate with seriousness of disease. I will say that having an envelope and not having an envelope does equate with the likelihood that a cleaner or sterilization or antiseptics or disinfectants will be effective. And so enveloped viruses are more susceptible to being killed. They're not as hardy. Non-enveloped viruses are harder to get rid of. They're more resistant to those cleaning mechanisms. All right, so now we're going to remind you guys of the lytic and the lysogenic phages, cycles. So this is getting into bacteriophages. These are viruses that infect bacteria. All right, so if you're a virus that infects bacteria, a temperate phage, I handle my pen too tightly, and it keeps popping up with things to change what I'm doing with my pen. Sorry. All right, temperate phage. Can switch between lytic and lysogenic phases. I'm going to talk about what they are in a moment, no worries. The lytic cycle is wherever we're multiplying. And the new viruses, the phages, will just multiply and multiply and multiply, and they will eventually burst out of the bacterial host cell. The lysogenic phase is where we will incorporate the prophage. When we're talking about this with bacteriophages, it's called a prophage into the bacterial host genome. And sometimes when that happens, if it brings extra genes with it, that code extra traits for the bacteria, then we would call those would be helpful to the bacteria. They would be called virulence factors. If it helps how bacteria survive, right? And if that happens, then this situation is called lysogenic conversion. All right. And with lysogenic conversion, this is one mechanism of horizontal gene transfer in bacteria. So remember, bacteria can exchange genetic information through horizontal gene transfer. They can do this through the sex pilus, where bacteria create a channel between one another and then shuttle DNA between them. They can pick up outside DNA, which was transformation, or they can have genetic information transferred by phage in this lysogenic conversion situation. And that is a process called transduction. So the process of transduction leads to lysogenic conversion, which is whenever you get extra traits when this lysogenic phase happens, introducing those new genes, those new virulence factors. All right, moving on. Yes, I will be posting these. Sorry, I just saw your notes on there, Alyssa. So yes, I will be posting these notes for you guys to study. All right. Genetics. Let's go over our genetic stuff. So now we've talked about viruses. Let's get into the details of our genetics. The genetic stuff that we learned about. First, we'll start with DNA replication. With DNA replication, we are literally talking about just making copies of DNA only. Just making copies of DNA specifically. So this, we have to understand a little bit about the DNA itself. So DNA is deoxyribonucleic acid. What is a nucleic acid? A nucleic acid is going to be like we call them a nucleotide, right? A nucleotide is a 5-carbon sugar of some kind, plus a nitrogenous base, plus a phosphate. If our 5-carbon sugar is deoxyribose, that's DNA. If our 5-carbon sugar is ribose, that's RNA, right? Our nitrogenous bases, actually, let me make little notes about this. Ribose equals RNA. Deoxyribose equals DNA. And then our nitrogenous bases are A, C, G, and either T or U. T is for DNA. U is for RNA. All right. So when we talk about replication, we have to understand a little bit about the structure of DNA, not just the nucleotides, which we just went over, but also how those nucleotides come together to create the DNA strand. And so our DNA strand, one strand, we usually would draw it like this, right? One strand. Where the backbone is alternating sugar phosphates. And the rungs are the nitrogenous bases. All right. And then when we get to the double-stranded format, we will have our backbone oriented one way for one strand, which we call 5' to 3', and then the opposite strand will be facing the opposite way. Well, and so things typically move in the 5' to 3' direction. So they're kind of opposite directions from one another. And this aspect of them being opposite directions we call that anti-parallel. Okay? And then, you know, if this was A, this would be T on the other side. And if this was C, this would be G on the other side. And so on and so forth with those guys. All right. All right, when DNA replication happens, you would imagine that we'd have to open up this whole double-stranded mess in order to replicate our DNA. So we're going to draw that. Stop moving. So we're going to have our strand, and I'm going to open it up. So here's our A's and T's and G's linked together. And at this spot here where we're going to open up, this is the replication fork. And here we have opening up. All right. So if this one is our 5 and this one would be our 3. So now here would be our 3 and now here would be our 5. So that we know which strand was which from up here. We've opened it up now. And basically you have to always, the enzyme that's going to add on, first it's going to use an RNA primer to start with, okay? But then it's going to add on to that RNA primer in the 5' to 3' direction. So adding from the 5', going to the 3'. This strand that can be made continuously, and we'll keep opening up that replication fork as we push into it. This one that is made continuously is called the leading strand. But as we open it up, let's say we opened it up a little bit, we could have added on a little primer in the 5' and then gone into the 3'. We can't make it this way, right? We can't do that. That is unallowed. We have to always move 5' to 3', always. So as we open it up, we'll add a little primer and add whatever's left. We'll open up a little more, add another primer, and then add whatever's left. And this one that is made in fragments, these fragments are called Okazaki fragments. And this is the lagging strand. These fragments will be made, again, in fragments by the DNA polymerase. So again, DNA polymerase is the enzyme that'll be trucking along. It started down here and trucked along into this direction. This is DNA polymerase. It started here and trucked along in this direction. DNA polymerase. Started here, DNA polymerase. Okay, so once we have our Okazaki fragments, we have to come in and seal them together because this fragment space needs to be sealed. We can't leave our DNA product in parts. And this is a problem because as we're going to be separating and transcribing things later on to make RNA and eventually proteins from our genes down the line, our DNA that we made, that we copied from the previous cell, has to be in one continuous piece. Otherwise, things don't work properly. So our fragments need sealing. And so the enzyme that does the sealing is called DNA ligase. All right. And so we can see with this process, looking at this, that one strand will be old. Stop. Oh my gosh. If we went from, this is our old, original, to now having separated the two old strands, and made copies of them. Excuse me. Through our DNA replication process that we just went through, that one strand is old and one strand is new. This is a process that is semi-conservative. We have half of it is conserved from the previous. It's old. Half of it is new. Half of it was not conserved. So therefore it is semi-conservative. This was some terms that people were getting very confused with on the previous exam, knowing the difference between what made it semi-conservative and what made it anti-parallel. All right, for Unit 2 exam, right? Hopefully this is coming back to you guys. All right, so we've made copies of our DNA, and now we can make a new cell and whatever. But in order to make a new cell, we're going to need new proteins in that new cell. And so we're going to need to go through transcription. So transcription is the process of going from a DNA template to making a RNA copy. So it's not the same nucleotides, right? From going deoxyribose to ribose, and not using thymine, but using uracil instead. So the steps involved, the first one is initiation. This is where RNA polymerase is this still recording? It says it's still recording. I don't know why I'm, whatever. RNA polymerase binds at the start, which is called something specifically. When we're going through transcription, the start is called the promoter. Okay. All right, so we bound there and we can open up our DNA, and now we can go through our elongation, which is our second step. This is where we're going to add complementary RNA nucleotides. I'll kind of walk you through it here in a second, that match the DNA sequence. And I don't forget where I am. Okay. So in DNA, we would have A, T, C, G, where A binds across to T, T to A, C to G, and G to C. It was just DNA. And this is thymine. And our different possible nucleotides are A, C, G, and either T or U. Adenine. These are nitrogenous bases. Okay. So depending on if you're making DNA or RNA, you'll use thymine or uracil. All right. So our RNA, A, U, C, G, where A binds with U and U binds with A. That's where our uracil is going to be. All right. Again, DNA uses the 5-carbon sugar deoxyribose, and RNA uses the 5-carbon sugar ribose. These are our differences. DNA is also double-stranded, and RNA is always single-stranded. It might fold back on itself, but it's only going to be one strand of RNA. So when we think of our different types of RNA, like tRNA and mRNA, which we'll talk about just a second, those tRNAs might fold back on themselves to make a weird shape, but it's still just one strand. Okay. So our elongation step, you would have your DNA template, And then your mRNA product, what we call, specifically, we call that product something. It's called a transcript. This is transcription. Going from nucleic acid to nucleic acid, we are just transcribing. We are rewriting it in our own handwriting instead of the, Smithsonian Institution's printed instructions. Now we've taken our own handwriting and made notes, still written, right? All right, so this DNA template might be A, C, whatever, T, A, G. And then our mRNA transcript would be U, A, G, A, U, C. Okay. Then our third step of transcription is termination. It gets to a specific sequence called the terminator sequence or termination sequence. Wow, my handwriting today. Okay, well, I clicked eraser. Which we didn't really talk about too much, and there's no details that we need to worry about with that one, okay? But those are the three steps: initiation, elongation, and termination. When we reach the termination sequence, the RNA polymerase falls off, and the transcript is released. Okay, so our RNA types. We have three types of RNA. First one is messenger RNA, the one we typically think of. It's the working copy of the gene. The second one is going to be ribosomal RNA. It is the structure of the ribosome, which is the machinery that will read it reads the mRNA to make the protein product with amino acids. Structure of ribosome. And the third one is tRNA. Transfer RNA, which carries amino acids based on whatever the mRNA sequence is. Why? Why? Okay, so we're going to go over translation next, which is using these three types of RNA to make a protein product. So that's our third step, is translation. We are now going from a nucleic acid language in mRNA transcript to making an amino acid-based language, which is the protein. So it also has three steps, and it's the same. They're called the same thing. The first one is initiation. The ribosome parts, there's two parts to a ribosome called subunits. And they will bind to the mRNA transcript. And they will start whatever they're going to be doing at the AUG codon. That's at, by the way. And that AUG codes for a specific amino acid called methionine. All right, so here's what it looks like. Do my best here. Make sure I give myself enough space to work. This is our mRNA transcript. A working copy of the gene, right? Our first codon, it would be AUG. Draw it up here. Right? And so then what happens is the ribosome comes and binds on. So let's say that's AUG. And I don't know what this one is. UUU. Who cares? I don't know all of my codons, so don't judge me too hard. Okay. So each set of 3 is known as a codon. 3 RNA nucleotides sequence. That's a codon. And a codon codes for an amino acid. So what happens is our ribosome will come in and bind onto this. And let's say that it's kind of got 2 going on. So we've kind of done the first one. This is what the first one would look like. It would have had UAC on its little tRNA. All right. And this would have been linked to an amino acid, which in this case is methionine, because AUG would match up. This is our codon, would match up to the anticodon, UAC. The anticodon is what is found on the tRNA. All right. And then that happens first, and then it would shift down to create this next opening. So this has already happened. We've already shifted it down to create this next opening. And so this one, be linked to a different amino acid. This would be AAA. And then these guys would create a bond. And this bond is known as a peptide bond. Right, and then this would shift down again to move this one over here to this spot and then kick out this tRNA and leaving that methionine linked onto that next amino acid, and so on and so on and so on and so on until we get to something like UGA or something like it, which would be signaling for stop. So stop codon causes the ribosome to fall off and release whatever polypeptide that it may, as we're linking these amino acids together and getting rid of these tRNAs they were linked to, opening up the next spot, and so on and so on. So this whole process, as we're moving down, this is called elongation. The tRNA anticodon matches the mRNA codon and carries a matching amino acid linked to the next one. by peptide bonds. A peptide bond, so if we looked at an amino acid, all amino acids look like this. We'll have NH2 and carbon, which will have a hydrogen on it, and some sort of thing that makes it that amino acid called an R group. So alanine has a certain R group, methionine has a certain R group, cysteine has a certain R group, glutamate, certain R group, right? And they all have this CO2 at the end. There's another H on there too, but don't worry too much about the H. Okay, guys? I know I'm not covering all my bonds, but basically the next one comes in and binds to the next NH2, CHR, CO2. And then this is one amino acid. And this is another amino acid. And this bond is the peptide bond, which you should have learned about in your GOB course if you took GOB, right? And then finally, when we get to whatever your stop codon is, that's the termination step. Stop codons are nonsense because they don't code for an amino acid. Because they don't code for an amino acid, this causes the ribosome to fall off, and that'll release that polypeptide product. All right, so that's translation. Nope. Genetics. I keep on track. Okay, so that's it for the genetics chapter. Next, I'm going to move on to the metabolism chapter. The first thing we're going to talk about are the enzymes. Not getting enough love from you guys. There are simple enzymes and there are conjugated. Simple enzymes, they're just the protein. And that functions just fine on its own. Conjugated enzymes are a little more complicated, and they're the ones that the chapter was kind of covering a little bit more. The whole functional conjugated enzyme is called the holoenzyme. The protein portion of this is called the apoenzyme. And the okay, so I want to, before I move on. For the protein portion, the apoenzyme, this is where the substrate will bind. The substrate is the thing the enzyme acts on. And that spot where it binds is called the active site. All right. All right, so then I don't want to change that. I want to erase this. So we can keep track of where we are. All right, so that's the protein part. The non-protein part of the holoenzyme, which the whole thing of why it's a conjugated enzyme is because it has a protein part and a non-protein part. If you're just protein, you're a simple enzyme. But if you need a non-protein part to function, like hemoglobin needs iron in order to function, right? So it is a conjugated protein. So conjugated enzymes act on substrates, bind the substrate, act the active site to change it, and that happens in the protein portion of the conjugated enzyme called the apoenzyme. But that conjugated enzyme can't be functional as a holoenzyme without its non-protein part. That non-protein part is always called a cofactor. Always. If you are organic, we will call you a coenzyme. It's a type of cofactor that is organic. If you are an inorganic cofactor, like iron, often metal ions are these, right? Then we just still call you a cofactor. Doesn't change. It's just a cofactor. But if you're an organic cofactor, you get a special name called a coenzyme. We have to be able to regulate our enzymes. We can do that at the genetic level, or we can do that by acting on the enzymes themselves. If we're going to act on the enzymes themselves, there's two different ways that we can regulate them. Competitive inhibition and non-competitive inhibition. If it's competitive inhibition, the inhibitor, whatever it is, will bind at the actual active site instead of the substrate, then blocking the substrate from being able to bind. It literally competes with the substrate. Non-competitive, we're already getting to 11:15. All right, non-competitive inhibition is where the inhibitor, whatever it is, binds somewhere else. It's not at the active site, but the site where it binds is either called the allosteric or the regulatory site. And that causes the enzyme to change shape. Typically so that the active site is no longer accessible. If that happens, this is not competitive, right? We're not competing with the substrate for the active site. We're binding somewhere else and then changing that enzyme shape so that the substrate can't bind at the active site. And so that's non-competitive inhibition. It's usually a product of whatever your process is. So when that product starts building up, it's a feedback mechanism to control your enzymes. So we learned a little bit about this with our operons, which I'm not covering in this review. If you want that reviewed, you guys can either go back and rewatch that or listen to one of the audio shorts on it, but I won't be able to cover it today. We have several different classes of enzymes, but there are four big ones that I really wanted you guys to be aware of. The first of the four biggies is the transferase. Transferases just move groups around from molecule to molecule. The second one is the ligases, like DNA ligase, right? We've already had a ligase. These join smaller parts to larger parts. The third one is the hydrolases, and we learned about hydrolysis in the lab. Hydrolases, like amylase or gelatinase or urease, what did they do? They broke larger molecules into small parts. And then the last one that we know and we love, the oxidoreductases. Yes, I know there were two other ones. So the oxidoreductases, before we get caught up in that, oxidoreductases are involved in electron transfer. Yes, so the other two that I am not as concerned about, just FYI, they won't be on your exam, were the isomerases, which change the orientations of molecules, and the liases, which are involved in double bonds. Don't care about them. Okay, moving on. All right. Also in this chapter were the redox reactions, real briefly, because this ended up being an important concept for understanding a lot of things further on down the road, right? So I want to be sure that we're clear on it. Again, redox reactions, reduction and oxidation, right? So these are going to be electron transfer always. If something is reduced, something else was always oxidized. They always come together. So what is oxidation and what is reduction? Oxidation is losing an electron. Reduction is gaining an electron. And the ways that you can remember it were Leo says GER or oil rig. And I'll write those out for you guys just so that we know. I keep bringing them up because they're important. I don't know why I wrote that a different color. It's going to bother me. Oil rig. L is lose. E is electron. O is oxidized. G is gain. Electron reduced. Oil is oxidize is loss. Reduction is gain. And these are really important concepts to understand with what we're going to be reviewing next, which is our aerobic respiration stuff. Because aerobic respiration, what happens at the end of the electron transport chain, the whole point of the electron transport chain in aerobic respiration, it makes ATP by oxidative phosphorylation. Sorry that got cramped up in there. Oxidative phosphorylation, which is ATP production that is driven by electron transport. All right, so you know what's coming next. Aerobic respiration. There's three major phases to aerobic respiration, which is glycolysis, Krebs, and the electron transport chain. For glycolysis, we are taking a 6-carbon glucose and turning it into 2 3-carbon pyruvates. The whole point of glycolysis is to get the pyruvate. It happens to be that we also, as a result of this, make 2 net ATP. We make 4, but we end up using 2 of them in the process. So it's 2 net ATP. And we also make some NADH, which is an electron carrier. It's a coenzyme, which we learned about coenzymes, right? They are organic things that work with enzymes to make a holoenzyme, right? So NADH is a electron carrier coenzyme. It's going to work with our other enzymes in the electron transport chain by carrying these electrons for it. The problem is that this is an electron here. It's an electron carrier. This H is bringing with it an electron. If you are a fermenter, you aren't going to be going through the electron transport chain. So in fermentation, we have to get rid of this. Otherwise, the electrons are building up and we're not going to be going through the electron transport chain. Why can't we go through the electron transport chain if we're a fermenter? Because we don't have oxygen to get rid of all those build up electrons and protons. Oxygen isn't there as our final electron acceptor. And that's the only one that it can use as a final electron acceptor if you are a fermenter. So instead of going through the electron transport chain, you just go through glycolysis. So you have to get rid of this electron. So what happens is to get rid of it, they feed it back into pyruvate. And fermenters will turn pyruvate. They'll feed that electron into pyruvate. And when you give an electron to somebody, you're changing their chemical structure because it's coming along with a hydrogen and maybe other things are happening too. Fermenters will turn pyruvate into ethanol, lactic acid, and or carbon dioxide, typically. But that's where they're making the ethanol, the lactic acid, and the carbon dioxide that we were testing in our biochemical tests. It's coming because they had to get rid of that electron, and they get rid of it by feeding it back into pyruvate because they don't need pyruvate for the Krebs cycle. They ain't going through the Krebs cycle. So they can get rid of pyruvate and get rid of that electron at the same time. And they make ethanol, lactic acid, or carbon dioxide as byproducts of that. So remember, in glycolysis, fermentation stays here. Really. So they're only going to make 2 net ATP. So all this red has to do with our fermenters here. All right. Next step is we have a little bit of an intermediate step. If you're getting into going into the Krebs cycle, we don't just use pyruvate. We will actually take our 3-carbon pyruvate and turn it into a 2-carbon acetyl-CoA. Coenzyme A is what that is. And this happens to give off a little bit of CO2 right here. This is the first time we're giving off carbon dioxide in all of this aerobic respiration process. And this is why, one of the reasons why you exhale carbon dioxide right here, because your pyruvate lost to carbon dioxide getting turned into acetyl-CoA. So the next step, the next big step is the Krebs. What happens here? We'll take that 2-carbon acetyl-CoA and feed it through our Krebs cycle, which you are welcome that I don't make you know all the steps in it from my class. And we'll cycle it. We'll add it onto a 4-carbon thing to make a 6-carbon thing. And in the process of the whole cycle, we'll kick off some stuff. One of the things we kick off is CO2. We know that. Carbon dioxide, a waste product. We don't need it for anything. Another thing that we'll kick off is 2-ATP. And another thing that we kick off, which is our goal for the Krebs cycle, keeps getting rid of my stars, is going to be NADH or FADH2. And what are these really? They're electron carriers, right? But the big take-home point of this, electron carriers, is the electron aspect. Because the next step is the electron transport chain. So, next step, electron transport chains. We've got a membrane. Here's our membrane. Phospholipid bilayer, whatever. Here's our little phospholipid tails. And here's the bilayer aspect. The tails are hydrophobic. The phosphates are on the outside. The tails are on the inside. And we got these little guys here that are kind of all, crushed up next to each other. And these are proteins, and these proteins have a name, and they are called cytochromes. This last one is called cytochrome C oxidase. Uh-oh. Okay, so there's cytochrome C oxidase. It's coming full circle, y'all. Okay, so here's some more membrane, whatever. And then these are all crushed right up next to the whole point. No. It looks kind of like this, whatever. And it's kind of a channel. So we'll kind of draw it like that. And this whole mess that I just drew here very beautifully is ATP synthase. And here's some more membrane, whatever. I had to make it massive, of course. So that's really, there we go. Okay. That's ATP synthase. And so if this is in bacteria, this is on the inner membrane, the cytoplasmic membrane, the cellular membrane, whatever you want to call it, the plasma membrane. If this is in eukaryotes, which remember they fall into the domain eukarya. If this is happening in eukaryotes, this is happening on the mitochondrial cristae, which are the folds inside of the mitochondria that probably used to be a bacterial inner membrane. Okay, so that's what this is. That's what this membrane is. That's where this is happening. Okay, so what happens is electron transport chain will make itself 34 ATP. That's what's going to be made. And this is the goal of the electron transport chain, is to make the ATP. And the way that that's done is we take the NADH or the FADH2, whatever it is, and we take off, let me change colors here. We take off the electrons and we take off the hydrogens. So they're protons because they lost their electrons. And then as a result, these give back to go do more of their stuff. NAD and FAD, they go back to pick up electrons from the Krebs cycle and crap like that. So, but what's important to us is that these electrons are going to shuttle through these cytochromes. And as the electrons are shuttling through those cytochromes, the H pluses that we've gathered are going to move out. So there'll be multiple of them from several FADs and NADs and whatever. So we're going to move these guys out. And we're going to build up a whole bunch of H pluses out here, which we'll just put pluses out here just for now. Put an H plus here so we can see it. So these protons build up out here, and this is called the proton motive force. And this process of the protons moving across and then now wanting to move back in, now that we've created a gradient, the protons are going to move back in because of diffusion. This process of diffusion is called chemiosmosis. We've created this gradient by moving the electrons across, forcing the protons out, and now the protons want to come in because of chemiosmosis, because diffusion, because we have a gradient, because we have too many protons outside and not very many inside, and they can only come in through this little hole through ATP synthase. Once we have gone through ATP synthase, of course, the whole point of the whole thing is to take ADP and then make ATP by adding a phosphate. And that this process here is driven by moving electrons. So we have now made ATP by oxidative phosphorylation. We put ox-phos. We talked about this in the previous thing, and so that's what is driving making ATP. This is the goal of the whole thing, is to make that ATP. But now we're building up protons. If we want to keep this thing going, we can't let the protons build up in here. Also, cytochrome C oxidase, it needs to lose its electron because oxidase, lose electron, oxidize, right? So it's an oxidase. So it wants to get rid of this electron at the end here. We get electrons here. So we'll take these electrons, we'll take 2 of these electrons from that process and 2 of these H pluses. And we'll take a nice deep breath in and get oxygen. And now you have made H2O. So oxygen is our final electron acceptor. So if we've gone from O to half of an O2, right? We've taken that O and we've added electrons in the form of the hydrogens. We have gained electrons. So therefore, oxygen has now been reduced. It has gained electrons. This is aerobic respiration. Now, with our guys that we talked about earlier in the lab, if our guys made H2S, they are using something else. They're not using oxygen. They're using something else, some sulfate product or whatever. So whenever they're using whatever they're using, they can make H2S. True, like anaerobes that aren't necessarily going through fermentation, right? Because fermentation will keep you in glycolysis, but you're going through anaerobic. Now we can make H2S as a product from that. And that's how they'll be doing that stuff. Okay. So it's all the same principle. It's just whether you're doing it aerobically, where oxygen is your final electron acceptor, or anaerobically, where something else is. One of those might lead to H2S production. All right. We're moving on to the next thing, which is immunology. Do we have any questions about aerobic respiration before I move on? Okay. Immunology stuff. Starting with the basics. Remember that all white blood cells, WBCs, are also known as leukocytes. Don't confuse leukocyte with lymphocyte. All white blood cells, all of them, are called leukocytes. There are two types of leukocytes: granulocytes and agranulocytes. The granulocytes are all of the fills and the mast cells. The fills include three different types: the basophils, the eosinophils, don't I know it, and the neutrophils. The neutrophils are the most numerous and probably the hardest workers of all of our granulocytes. Right, the agranulocytes, we have the lymphocytes. There's only two types that we had to learn about. Those were the T and the B cells. And then we have the monocytes. When they're in the blood, they're called. If they move into the tissue, they're either macrophages, which I'll abbreviate like this, macrophage, or dendritic cells. Typically, the skin ones are the dendritic cells, and the macrophages are in the other tissues. All right, so this is general concept of those types of cells. We also have some cells that can act as phagocytes, and some cells that can act as, when we get into our adaptive or selective immunity, are antigen-presenting cells. Cells that can act as phagocytes are our neutrophils and our monocytes and their derivatives, including our macrophages and dendritic cells. Our antigen-presenting cells include our monocytes and their derivatives. And also, interestingly enough, B cells. Something I wanted you to notice here. Neutrophils, not antigen-presenting cells, but they are phagocytes. B cells, they are antigen-presenting cells, but they are not phagocytes. So how do the B cells present antigen if they're not eating it up like a phagocyte? Because they're B cells, remember a B cell has on its surface something that looks like an antibody. Do you guys remember this? This is its receptor. It's B cell receptor, which also happens to be IgD. It binds to a thing, takes this whole thing inside. Let me do a different color. maybe it'll like that better. Takes this whole complex inside, and then after that's gone inside, because it picked it up from outside, it'll present that on MHC2. It did come from outside. And we're going to get into this in just a second. We're going to review all this. But that's how B cells pick things up with their receptor. They're not phagocytosing anything. They are just using the receptor to bring things inside. So they are not a phagocyte, but they are an antigen-presenting cell. Antigen-presenting cells, specifically, antigen-presenting cells present outside antigen on MHC2. They're the only ones that can. And that's why they're called antigen-presenting cells, because they present antigen on MHC2. How do our neutrophils decide to phagocytose something? Because they're recognizing PAMPs, not antigens. What's the difference? A PAMP lets you recognize that that was a bracelet. An antigen tells you that this is that specific bracelet that Professor Yost was wearing that day. Very specific. Antigens are very specific to that pathogen. PAMPs just tell you this is not self. So an example of a PAMP, a pathogen-associated molecular pattern, would be lipopolysaccharide, aka endotoxin. We can see lipopolysaccharide, which is present on all gram-negative bacteria. If we see that, we know that's not self, eat it. If we see double-stranded RNA, we know that's not self, like we said earlier, eat it. If we see peptidoglycan floating around, we know that's not self, eat it. We don't need to know that it is Mycobacterium tuberculosis yet. Our innate system doesn't need to know that. But once we have eaten it up and broken it down, you can poop it out, and the macrophages can pick up what's left over and present it on MHC2 because we picked it up from outside. And now our T cells can say, this isn't just not self. This is from Mycobacterium tuberculosis or COVID or whatever. So that's what's really going on with all of that. And that's what the difference is. Okay. All right, so let's go over MHC. MHC stands for Major Histocompatibility Complex. Histocompatibility because it's also what we recognize as self. Histo is tissue, compatibility, compatible with self, okay? So it's a complex that also lets us recognize self and differentiate it from non-self. So there's what MHC does, MHC always, this is all it does, presents antigen specifically to T cells only. That's all it does. It is a little hand that holds out antigen for T cells to look at it. There's two different types of MHC, MHC1 and MHC2. MHC1 presents inside stuff that was found inside, not just stuff, antigen. Presents antigen found inside of cells. So it's usually infected cells or abnormal in some way. So therefore, all nucleated cells have MHC1. So all nucleated cells can call out for help if something is wrong inside of them. MHC1 is only recognized by CD8. CD8 is not a T cell. CD8 is not a cell. CD8 is a marker on the surface of cytotoxic T cells, CD8 positive T cells. Okay, so CD8 is a marker that recognizes MHC1. MHC2 presents antigen found outside of cells. Only antigen presenting cells have MHC2. And this includes our B cells, our monocytes, our macrophages, and our dendritic cells. They're the only ones that have MHC2. And then when we're presenting antigen on MHC2 is only recognized by CD4. Again, CD4 is not a T cell. CD4 is not T helper cells. CD4 is a marker on T helper cells. Okay. Are we ready? Because we're going to do T cells and then we're going to do B cells. And then I'm probably going to be done. I'm not going to lie. Okay. T cells. This is our T cell response. Here we have virus or whatever. Bad guy outside. An antigen presenting cell that is also a phagocyte can come and eat this up. In this case, I'm going to say it's a macrophage. This macrophage came and ate up. Come on, I don't want to play this game. This macrophage came and ate up this virus, digested it, and found it outside. So this is MHC2. I'm going to use the number 2 here. And I don't care if you use the Roman numerals or the number 2, whatever you feel comfortable with, okay? As long as I know what you're talking about, that is way more important to me. Okay, so here's our antigen from whatever we picked up outside. Maybe it's the spike protein from COVID. Who knows? All right, so if we are presenting this on MHC2 that we picked up from outside, we talked about MHC2 presents antigen from outside, and only antigen-presenting cells have MHC2. This was our macrophage presenting it. And what recognizes MHC2? CD4. So we know what's going to recognize it. Oh, not what I meant to do. I guess we could do that. It's ugly, but we'll do it. I don't like how bright the screen is. So this molecule here is going to recognize that. And that molecule is on another cell. So what cells have? We know what this is, right? We know because it's recognizing MHC2, this is CD4. It doesn't recognize the antigen. It just recognizes the MHC2. We know that. So what cells have CD4 on them? T helper cells. This isn't a Th1. This isn't a Th2. This is just a T helper. We call this a naive. It hasn't been told if it needs to be Th1 or Th2 or anything yet. It hasn't seen this antigen before. It is naive. It is just a T helper cell. So how does it see the antigen itself? It recognizes this specific antigen, very similar to how antibodies react, using the T cell receptor, which is a lot like the antibody-like receptor on the B cells, okay? But it's just on T cells and they don't secrete it. It's only on its surface. So the T cell receptor sees the antigen. It doesn't see it as foreign. It sees it as, this is the COVID spike protein. It knows exactly what it is, and that reacts very specifically to that. All right. So we have done that. MHC2 is presenting that outside antigen. Also known as exogenous, right? The T cell receptor sees that. Here we have the CD4 recognizing that it is MHC2 presenting it. Everything's happening like it should. So now what happens is that these guys can cross-activate one another. And so how does it do that? It sends activation signals in the form of cytokines. Great. Once this happens, once we've had this interaction take place, the T cell receptor sees the antigen. CD4 recognizes that MHC2 is presenting it. Now our T helper cell will proliferate. Actually, let me move that. And turn into T helpers. Just a whole bunch of T helpers. They're not special yet. Maybe this one becomes a T helper memory cell. This one might become a T helper regulatory cell. This one might become a TH1, and this one might become a TH2. This process here is the differentiation. That's what proliferate and differentiate means, really, when we say it, okay? All right. These are still T helper cells. So what's on their surface? They still got the janky old CD4. These are CD4 positive T helper cells. They're still the same. It's just now the TH1 cells. These are our captains. These are our effector cells that do the jobs. These guys are what are going to tell the rest of the immune response that they can do whatever they need to do. So we'll talk about TH2 in a second because I'm about to talk about the B cells next. But the TH2 guys regulate the B cell outside response. So B cells, antibody, and this is all outside stuff. Response stuff. Okay. The TH1 cells, what they're going to do is that separately, we got to think about this now. You have a lung cell. Out here on the side, and it got infected inside with a virus. Let's say it's COVID. I don't know. I hate that it does this. If it's infected inside, it's going to present this crap on MHC1, right? Came from inside, that goes on 1. Help, I'm infected inside with something. My inside, something is wrong in my inside. That's what's happening here. What recognizes MHC1? MHC1 is only recognized by CD8. So now what happens here is color that in a little better. I don't like it bothering me. Whatever, it's beautiful. Okay, so this is CD8, we know that. And where's CD8 found? Cytotoxic T cell, right? And how does it see the specific antigen? The T cell receptor. Again, the T cell receptor sees the specific antigen. CD8 recognizes MHC1. But it cannot do anything. Cannot do anything until it gets the go-ahead from its captain. So once the captain says go, it can do something. And so what's its captain? The TH1 cell, specifically that got activated from that outside earlier. TH1 activates that. Now that the cytotoxic T cell has been activated, it can release granzymes and perforins. That activation allows granzymes and perforins from the CD8 cell. Cytotoxic T cell. And then those lyse the lung cell. So that is the T cell response. So let's go over our B cell response. Somewhere out in the wild, in a lymph node somewhere. We have a B cell. And it is naive. It is not a plasma cell. It is not a memory cell. It is a B cell that's just waiting in the lymph node to see the one antigen that it was made to recognize. And how does it recognize that antigen? With its immunoglobulin receptor, the B cell receptor, which is also known as IgD, picks up antigen that is outside. Okay. If that then brings it in, and presents it. It came from outside, so it's presented on MHC2. This should look familiar. Yeah, we just did this with the macrophage. So what sees MHC2? CD4. Where is CD4 found? T helper. And in this case, this is a TH2. Why is it a TH2 already? Why is it not just a helper? What could be a naive helper? If it was a naive helper, it would have to go through all of this again. All of this stuff that we went through up here. They would have to go through all of this if it's a naive helper. But if we've already activated it from our macrophage in another part of the cell, we don't necessarily have to reactivate it. So in this case, we're saying that it got activated previously. If it hadn't gotten activated previously, the B cell can activate it just like the macrophage did up here. It's just that this reaction up here that created our naive helper to turn into the Th2, we'd have to wait on that because only Th2 cells can actually activate the B cell to do its thing. So this Th2 cell has been activated previously. It'll activate the B cell. Now our B cell will proliferate into a bunch of Bs, B, B, B. That's it. And then each of these Bs becomes either a B memory, a B regulator, or a, you've seen them. They look like this. This is endoplasmic reticulum, by the way. This is our effector B cell, also known as a plasma cell. All right. And so this is our initial first one that we just made. So this plasma cell, it'll make antibody. The first antibody that it makes will be IgM. Then we'll go through a process of class switching. Into either IgG, IgA, or IgE, depending on what signals the body is sending from the infection. Depending on what signals we're getting. And what are those signals called again? They're called cytokines. And so this is the response that we will be seeing going on with our antibodies. Move this a little bit so it's easier to see what's going on. So we know that our IgG is our workhorse antibody. It's found in the blood. It can be found in some tissues as well, but primarily in the bloodstream. And it can do all of the things that antibody can do. IgA, this is one that's found in the mucosa. We call it the secretory antibody. And it'll mostly just hang out in the mucosa for defense to protect the body from stuff getting in that way. And then IgE is usually associated with allergies or with parasites. And so we dictate whether we should make more or less of each of these types based on signals that we're getting from the body in response to whatever the attack is that's going on in the area. And then our memory B cells usually will make whatever one we switched to in high numbers. Okay? Any questions about any of this stuff? All right. So I was planning on doing the meta review, but this has gone pretty long. It's already gone almost, it's gone an hour and a half just from this. So I'm not going to do the meta review. I know that you think that it sucks, but I was just going to do the same exact thing that I did last semester. So I am going to post, the recording is already posted, but I'm going to repost it to remind you guys to watch it. It is a recording from last year of me doing the meta review. So I'm going to post that recording. So please watch that recording if you want to see me go through this stuff. But basically, I start with an HIV infection. I walk you through the immune response, but then I talk to you a little bit about, okay, if HIV has gotten in the cell, What does HIV need to do to make copies of itself? So I walk you through transcription and translation. I also walk you through reverse transcriptase and how that would work with the HIV. I talk about the HIV's genome and how reverse transcriptase works to make the DNA and insert itself into the human cell. If it inserts itself into the human cell, that might sound like lysogenic conversion, but we are not bacteria. We do not go through lysogenic or lysogeny or have prophages. We have proviruses. And so whenever that happens, we call that latency in our cells. So I'll talk through HIV with that, and I talk about how ELISA works to detect anti-HIV antibody. And if we're detecting an antibody, what type of ELISA would that be? That would be indirect. Then I will go through what happens with a staph skin infection. Instead of a viral inside infection, what happens with an outside infection, and some of the tests we might use to diagnose that staph skin infection. I'll talk about capsules. I'll talk about how antibody will help protect against staph and the different functions of antibody. That will be covered in that review. I will also talk about an outbreak, and I'll go through some of the epidemiological stuff when we discuss that outbreak, and that will be covered in that review as well. I have everything printed out here, so I'm trying to make sure you know what you're going to get in this when you watch it. I'll talk about diagnosing etiological agents with immunological tests and things like that for that outbreak that happened. And what our treatments might be once we diagnose that outbreak, what treatments we might use to approach that, and then go over the different antibiotics. And then lastly, in that review, I do go over operons, and I believe it's the last thing that I will cover in that review. So look out for that. So I will post that again. It's already in your extra final study review materials, but I'm going to post it again with these notes. So I'll post the link to this video. I'll post these notes. I'll post the notes for the meta review and the link to the video for the meta review, so you guys can watch that if you want to and start thinking about bigger picture approaches to these things, okay? Do you guys have any last questions for me before I go? All right, if that is it, thank you guys for coming today. I'm going to call it a day, and I'll post those other resources for you so you can refer to them. And good luck studying. Good luck on the exam. It's been a genuine pleasure this semester. And I will see you guys tomorrow at 10 a.m. And good luck on the exam. Yeah. Happy studying. Don't forget to get some sleep.